Hello guys, this is zoologist AK and I'm here to talk about regulation of gene expression in case of prokaryotes, in which I'm going to talk about trip operon, and this is my second part of trip operon, in which I will talk about the control of trip operon by attenuation. And in this video I'm going to include all these points. First is my what does this attenuation mean in E. coli, and second is how this attenuation process takes place, and third point is how this attenuation was first revealed, means how this attenuation process was first identified, how this repression and attenuation respond in same way to regulate the expression of different structural gene in case of trip operon, and attenuation can be controlled by translation also, which we are going to talk in this part of my topic. And which structure of ribosome determine that termination is attenuated means termination is suppressed only in the absence of tryptophan. How widespread is the use of attenuation means widespread use of attenuation means here that either this attenuation control is applied only in the trip operon or in others operon also. So we are going to talk about this also in my video. And last point is E. coli and Bacillus subtilis use the same type of mechanism to control the formation of mRNA polypeptide. So these are the points which I am going to include in this part of my video. As I am talking about this word attenuation, what does this word attenuation means here? In general term, attenuation means to reduce something, and in case of trip operon, attenuation means to reduce the rate of transcription. And before understanding the actual mechanism of this attenuation process to control the expression of different structural gene in case of trip operon, we need to understand the actual mechanism of gene expression. How does this gene expressed? Gene expression means the formation of functional gene product, and these functional gene product could be proteins or non-protein coding molecules. For example, tRNAs and small nuclear RNAs, these are non-protein coding regions, and mRNA here is our protein coding molecule. And this gene expression is summarized in the central dogma. This picture showed here the mechanism of central dogma, which was first formulated by Francis and Crick. It suggests that DNA has all the information to make proteins, and RNA formed as mRNA from DNA and code this information to the ribosome, that is the protein factory of the cell. And in ribosome, this information is translated from codes that is encoded by mRNA into functional polypeptide chain. As you can see here, this is a functional polypeptide chain, and these are the different types of amino acid in this polypeptide chain. And this expression of gene occur in two stages. First is our transcription, and second one is our translation. And as you can see here in the picture, that DNA will make new DNA molecule through the process of replication, and a new mRNA through the process of transcription. This mRNA along with ribosome, that is the protein factory of the cell, and tRNA that will transfer amino acid to the ribosomal peptidyl subunit to form a polypeptide chain, result in formation of protein through a process known as translation, and result in the formation of polypeptide chain. Now come back to our main topic, that is attenuation, that control the expression of structural gene in case of trip operon in E. coli bacteria. Here this attenuation attenuation means the reduction of rate of transcription of this different structural gene, and this attenuation occur at two levels. First level of control by tryptophan, because tryptophan amino acid will bind to the repressor molecule, due to which there will be a conformational change in the repressor, and this repressor will bind to the operator, due to which RNA will not be able to bind to the promoter site, and as a result transcription will stop. And second level of control is attenuator control, and it occur in the region of 5' leader molecule of mRNA chain. Here this attenuator contain ORF, open reading frame. This open reading frame is nothing but these are the length of DNA and RNA molecule which transcribe into RNA polypeptide chain that code for different amino acids and synthesize different types of enzyme molecules in the cell. It is a continuous this ORF open reading frame is a continuous stretch of codons that begin with a start codon, that is our AUG, and stop at the and ends at a stop codon that can be UAA, UAG or UZA, and ATZ stand here for the transcription start side codon. And this transcription termination site that causes the stop of transcription in case of E. coli bacteria during trip operon transcription process. This termination site is located after ORF open reading frame beyond its stop codon. This attenuator open reading frame allows E. coli to monitor the second pole of trip tRNA, as we know that this trip tRNA regulate this transcription process in case of E. coli. So high level of trip tRNA will attenuate. Attenuate means stop or reduce the transcription or terminate transcription in case of trip operon. And low level of trip tRNA allow the trip structural genes, that is EDCB and A operon, to transcribe into different structural molecules or different mRNA molecules. Now come to our second point, that is how does this attenuation process takes place. This attenuation process takes place by change in the secondary structure that is represented here, and change in the secondary structure of attenuator mRNA that formed by transcription of this attenuator region that is present in the trip operon DNA molecule. And this change in secondary structure of this mRNA result in the formation of hairpin loop structure, and this hairpin loop structure cause attenuation process that will reduce the rate of transcription. So here ribosome create a mechanism that regulate the translation of this attenuator region, and this attenuator result in formation of hairpin loop structure, and this hairpin loop structure result in attenuation or reduce the rate of transcription. And when there is the low level of this strip tRNA molecule in the cell, this ribosome will not translate this attenuator and leader region, due to which termination hairpin structure or terminator hairpin structure will not form, due to which RNA polymerase will able to bind to this promoter site and result in the transcription of these different structural gene into different enzymes. Now come to our third point, that is how does this attenuation was first revealed. This attenuation was revealed by deletion of sequence between the operator and trip E, as you can see here in this picture, here is the operator site, here is the trip E, and the sequence that was deleted in between both of them is the sequence that code for leader and attenuator mRNA chain that result in formation of hairpin loop structure that cause attenuation process or reduce the rate of transcription in case of trip operon. And this operator is the binding site at which the repressor molecule will bind, and trip is the structural gene of this trip operon. And due to deletion of the sequence in between the operator and trip E result in increase the expression of the structural gene, and this expression of structural gene was independent of repressor molecule, and both the level of transcription increase, and these level were basal level of transcription and derepressed level of transcription. Derepressed level of transcription here mean in the absence of tryptophan molecule, as we know that tryptophan molecule bind to the dimer of repressor, due to which conformation change in repressor occur, and repressor bind to the operator and inhibit the binding of RNA polymerase to the promoter, due to which transcription of different structural gene is suppressed. So there was the increase in transcription at both the level of basal level and derepressed level. So this site that was deleted was called attenuator, and to date also we call it attenuator because it result in the attenuation or affect the events of RNA polymerase, because RNA polymerase bind the promoter and start the transcription of different structural gene in case of trip operon. Now come to our fourth point. Repression and attenuation respond in the same way. Same way means here they regulate the transcription of different structural gene in case of trip operon in the same way. So first we will talk about mechanism of repression. Here we will study about the role of repressor molecule. As we know that when tryptophan level is high, the free tryptophan amino acid bind to the dimer molecule of repressor and result in conformational change of this repressor molecule, due to which this repressor bind to the operator site and inhibit the binding of RNA polymerase to the promoter. As a result, transcription of different structural gene of trip operon is inhibited, and when this trip operon is level is low in the cell, then RNA polymerase is freely bind to the promoter site, result in transcription of different structural gene. And now come to the mechanism of attenuation. When tryptophan level is present, it result in negative 10 multiple effect on transcription, means it reduce the rate of transcription. But when this tryptophan is absent, it result in negative 7 multiple increase in initiation of transcription by RNA polymerase. It result in 700-fold of increase in regulation of trip operon that is caused in the presence of trip operon. So it result in 700-fold of increase in regulation. So this was so from this we get to know that the repressor molecule and this attenuation mechanism play similar role in the inhibition and activation of transcription of different structural gene in case of trip operon. Now come to our fifth point, that is attenuation can be controlled by translation. How does this translation affect attenuation process? This translation result in the translation of leader peptide that is about 14 amino acid long into a polypeptide chain, and this secondary structure of this polypeptide chain will result in the formation of hairpin loop structure. This hairpin loop structure configuration into different alternating base pair configuration result in the formation of attenuation or reduction in transcription of different structural gene in case of trip operon. In this picture you can see here that the leader region is transcribed into a short polypeptide chain that is about 14 amino acid long by RNA polymerase, and this polypeptide chain is translated by ribosome, but the translation of this leader peptide will stop when ribosome will reach up to the trip operon, and this leader region consists 3 part. One is ribosomal binding site, and second is start codon, that is AUG, and third is two successive trip codon. And the translation of this leader region will stop when the ribosome will reach up to this trip codon. So this trip codon play a crucial role in leader peptide coding sequence. And this sequence of mRNA that is formed by translation of this leader peptide suggests that ribosomal stalling of this translation of this leader region at trip codon influence the termination at the attenuator or attenuator site. It influence the termination of translation process. To understand this in detail, look at this picture. This picture showed the trip leader region that exists in alternate base pair conformation. And you can see here in this diagram, this region in the middle shows that four region of this trip leader that could base pair with each other. And to understand this termination or intrinsic termination, look at this diagram. Here you can see that region first pair with second region, and this is in this diagram you can see this is the first region, second region, and here first and second regions are present in pairs, and region third is paired with region fourth, due to which a hairpin loop structure generate that proceed the U8 uracil 8 and result in the formation of hairpin loop structure. This is the essential signal for this termination or called intrinsic termination. And the RNA chain of this leader region form this structure automatically. When this region first is prevented to form pair with this region second, then a different type of structure is formed, that is shown in this diagram here. As you can see, in this case region 2 is paired with region 3, and region 4 has no available pairing pattern, so it remains in single stranded conformation, due to which the terminator hairpin loop structure doesn't form, and it does not result in intrinsic termination at the attenuator, or it does not terminate the process. Now come to our sixth point, that is which structure of ribosome determine that termination is attenuated only in the absence of tryptophan. The position of the ribosome can determine which structure is formed in such a way that termination is attenuated, means termination of transcription is stopped only in the absence of tryptophan, which is explained in the diagram given below. As you can see here in this picture, that when tryptophan is absent, ribosomal stall at the trip codon, means the translation of the leader region or leader peptide will stall or stop at the trip codon that is present in the leader region of trip operon, and these trip codon are part of region 1. As you can see here in this picture, this was our region 1. So these trip codons are present in the region 1, and these result in stall of translation of the leader peptide. So when this stallation process occur, this region first of this trip leader will not base pair with leader second, that is shown in yellow color, and due to which this second region result in the pair with the region third, due to which the region fourth, that is shown in light blue color, will remain in a single stranded form. In the absence of terminator hairpin, so it will not form hairpin, and in the absence of this hairpin loop structure, RNA polymerase will continue transcription of different structural gene bypassing the attenuator site, because as we know that here is the promoter site, then our leader region and attenuator site come, and then our structural gene lie. So this ribosome will bind to this promoter site and pass this terminator site when the tryptophan level is low, and result in the transcription of these different structural genes. And when there is the high level of tryptophan or the high level of tRNA inside the cell, ribosome are able to synthesize the leader peptide into a polypeptide, or will translate this leader peptide. And as a result of continuous synthesis of leader polypeptide due to this ribosome, that this translation process will stall at UGA codon, that is the stop codon, which lie between the region first, that is shown in light pink color, and this region second, that is shown in yellow color. And by processing this point, the ribosome extend over region 2 and prevent it from base pairing with the region second, and result is that region third is available to base pair with fourth region, which generate a terminator hairpin loop structure in this condition. And as I told that our promoter is present first, then our leader region, then our terminator region, and then our structural genes are present in case of tryptophan. So this ribosome, this RNA polymerase has to cross this terminator side, but when the hairpin loop structure is formed due to translation of this leader peptide, then it is unable to transcript these different structural gene, and the and RNA polymerase terminate at the attenuator site. So this explain how can termination of transcription means stop of transcription at the attenuator site, or attenuator site that is present between the promoter region and the TRPE, that is the first structural gene, respond to the level of tRNA molecule level inside the cell. But the control of attenuation site that is present between promoter and the structural gene, this attenuation site control require a precise time of event, means it occur at precise time, because ribosome movement to determine formation of alternate secondary structure that control termination, that is the formation of hairpin loop structure. Translation of leader must occur at the same time when this RNA polymerase result in the synthesis of polypeptide. So this translation of this leader polypeptide and transcription of this leader polypeptide should occur at the precise time, that then only this hairpin loop structure will form and result in the termination of transcription, and will stall or will stop the RNA polymerase at the attenuator site. And the RNA polymerase remain posed at the promoter site. This is the promoter site, and RNA polymerase will posed at this promoter site till the leader peptide that is formed by transcription of the attenuator and leader region. This leader polypeptide, up to which this leader polypeptide is not translate, and the polymerase is then released and move off towards the attenuation site, A site or attenuation site. And by the time it arrives there, the secondary structure of this attenuation, that is the hairpin loop structure, determine whether this RNA polymerase will bypass this hairpin loop structure or not, and will synthesize or will express these structural gene by transcribing them or not. Now come to our this point, how widespread is the use of attenuation as a control mechanism for bacterial operon. It is this attenuation control used in at least about 6 operon that code for enzymes that are concerned with the biosynthesis of different amino acids. So this attenuation mechanism is widespread. And another point is E. coli and Bacillus subtilis use the same mechanism of control. Yes, they use same mechanism which involve control of mRNA structure in response to the presence or absence of transfer RNA, but they have combined the individual interaction in different way. But the end result is same in case of E. coli and beta or Bacillus subtilis. To inhibit production of the enzyme when there is an excess supply of amino acid. For example, in E. coli, this excess supply of tryptophan inhibit the production of different enzymes. And to activate production when a shortage of these amino acid is present. And to activate production when a shortage is indicated by the accumulation of uncharged transferred tRNA tryptophan molecule. So this was all about the control of trip operon by attenuation process. Hope you all get to learn how does this attenuation process control the trip operon. If you have any query, please write in the description box given below. And please do like, share and subscribe to my channel. Thank you so much for your patience and for watching my videos.